Welcome friends to a new session on molecular biology. In this session, we will deal with organization of eukaryotic and prokaryotic chromosomes. So far, we have been dealing with DNA, RNA, but don't you think that if they are in highly organized inside the cell or nucleus, cell have been translating, transcribing or replicating throughout its life meaninglessly. This mystery dissolved by the discovery of chromosomes. Cell wraps the whole genome well and good, protected and make available for them if and only if the situation demands. Discovery of chromosome. The word chromosome means colored bodies. It contains the cell's genetic material which determines how an organism develops. Although Mendel provided convincing evidence that inherited traits were governed by discrete factors or genes, but his studies were totally unconcerned about the physical nature of these elements or their location within the organism. With the discovery of DNA in 1953, one of the greatest breakthroughs made it possible to express the biological concepts and structures in concrete chemical terms. The rediscovery of Mendel's work and its confirmation had an immediate influence on research in cell biology. Whatever be their physical nature, they have to be inherited in a consistent manner with Mendelian principle. The term chromosome was first suggested in 1888 by a German anatomist, Heinrich Wilhelm Gottfried von Waldeier-Hartz, 1936 to 1921. He used the term to describe certain structures that form during reproduction. In 1903, Walter Sutton, a graduate student at Columbia University, published a paper that pointed directly to chromosomes as the physical carriers of the Mendel's genetic factors. Sutton followed the development of sperm cells in the grasshopper because they got large and easily observable chromosomes. On examination of spermatogonia, Sutton counted 23 chromosomes and suggested that they are present in look-alike pairs. Sutton soon realized that they are organized in pairs or called them as homologous chromosomes and correlated perfectly with the pairs of inheritable factors uncovered by Mendel. What are chromosomes? Chromosome is a single piece of coiled DNA containing many genes, regulatory elements and other nucleotide sequences. The word chromosome comes from the Greek word chroma, means color, and soma means body. During interphase, when cells are not dividing, the genetic material exists as a nucleoprotein complex called chromatin, which is dispersed through much of the nucleus. Further folding of the chromosomes occurs in the mitosis, producing visible metaphase chromosomes. Now, chromosome morphology. Early microscopic observations on the number and size of chromosomes and their staining patterns led to the discovery of many important general characteristics of chromosome structure. Each metaphase chromosomes consist of two sister chromatids, which are attached to a centromere. The location of the centromere on each chromosome gives a chromosome its characteristic shape and can be used to help describe the location of specific genes. This figure shows the schematic representation of chromosome inside the cell. Each chromosome has one centromere with one or two arms projecting from the centromere and a telomere. A centromere is visible as a primary constriction on mammalian chromosomes. In humans, this site is characterized by the presence of a particular type of tandemly repeated symbol DNA sequence called alpha satellite. Alpha satellite DNA is a 170 base pair sequence repeated over many millions of base pairs of DNA. Telomeres are specialized structures at the ends of chromosomes. Telomeres hide the ends of the chromosome from the mechanism within the cell that monitor DNA damage. They are also needed to overcome the problem of end replication. Chromosomes are classified according to the centromere position. First one, metacentric. Metacentric chromosomes are V-shaped, and in these chromosomes, the centromere divide roughly into two equal arms. In the case of amphibians, it have metacentric chromosomes. Second, submetacentric chromosomes. In submetacentric chromosomes, centromere is submedian, giving one longer and one shorter arm. The submetacentric chromosomes are L or J shaped during anaphase. Third one, acrocentric. In acrocentric chromosome, the centromere is located terminally and forms two unequal arm lengths. The locust acrididae have the acrocentric chromosomes. The P arm is too short to observe and they appear rod shaped during anaphase. Please see the figure on screen. It shows the morphology of chromosome according to the position of centromere. Fourth one, telocentric. In telocentric chromosome, the centromere is located at one end. They appear rod shaped during anaphase. Now, the cell cycle. The diploid cells of our body are derived from the original diploid fertilized egg by repeated rounds of mitotic cell division. Each round can be summarized as one turn of the cell cycle. This comprises a short stage of cell division, the M phase, that is mitosis, and a long intervening interphase. Interphase can be divided into S phase, DNA synthesis, G1 phase, gap between M phase and S phase, and G2 phase, it is a gap between S phase and M phase. From anaphase of mitosis right through until DNA duplication in S phase, a chromosome of a diploid cell contains a single DNA double helix and the total DNA content is 2C. G1 is a normal state of a cell and the long-term end state of non-dividing, that is spacing cells. Cells enter S phase only if they are committed to mitosis. Non-dividing, that is spacing cells, remain in a modified G1 stage, sometimes called as G0. Please see the figure on screen. It gives the different stages in cell cycle. Interphase chromatin. During interphase, that is the period of the cell cycle where the cell is not dividing, two types of chromatin can be distinguished. The density of the chromatin that makes up each chromosome, that is how tightly it is packed, varies along the length of the chromosome. During interphase, most of the chromatin, that is euchromatin, exists in an extended state, dispersed through the nucleus and staining diffusely. While staining, heterochromatin stains denser than euchromatin. Please see the figure on screen. It describes the chromatin condensation changes during the cell cycle. Euchromatin. Genes located in this region may or may not be expressed depending on the cell type and its metabolic requirement. Heterochromatin, either due to naturally or chromosomal rearrangements, are very unlikely to be expressed during these chromosomal stages. Heterochromatin can be further distinguished into two types. Constitutive heterochromatin. This is always inactive and condensed. It largely consists of repetitive DNA and is found in and around centromere. Facultative heterochromatin. It can exist in either genetically active, that is decondensed, or inactive, that is condensed form, as in the case of mammalian X chromosome inactivation. This figure shows the euchromatin and heterochromatin. Individual chromosomes cannot be distinguished at this stage. They appear in nucleus as a homogeneous tangled mix of DNA and protein.